Hey guys, so this video is going to go over Lab 8, which is about cell division in eukaryotes. So to start off, we have somatic cells and gamete producing cells that enter mitosis. But before they enter mitosis, they are both in interphase. During interphase, our cells are going to grow, perform their normal functions, and then duplicate its DNA in preparation for cell division. Interphase has three different phases. We have G1, which is the growth phase. We have the S phase, which is DNA synthesis. And then we have that G2 phase in which the cell is going to prepare for division. Our somatic cells, they go through interphase and then they go into mitosis to produce two identical daughter cells. Our gamete producing cells, so our germ cells, they go into mitosis. But these diploid cells, they also go through interphase. And just like in mitosis, the DNA is going to be duplicated during the S phase. Then it's going to go into meiosis 1 and 2, and it's going to end up producing 4 haploid gametes, either sperm or eggs. So even though mitosis has 2 rounds of division, DNA is only going to replicate once during interphase before meiosis 1. So to summarize, all dividing cells have to go through interphase first to prepare, whether they are going into mitosis or meiosis. So in mitosis, in G1, the cell is going to grow in size and produce proteins and organelles that are needed for normal function. This is the longest part of the cell cycle. The nucleus is going to appear large and lightly stained under the microscope with no visible chromosomes. So we're going to have only chromatin, which is uncoiled DNA. During the S phase, which is synthesis, we have DNA replication. So each chromosome, it's now going to consist of 2 identical sister chromatids. The cell is still performing its regular duties, but behind the scenes, it's duplicating its entire genome. Then we get to G2 phase, which is gap2. The cell is going to continue to grow and prepare for division by producing the proteins and enzymes that are needed for mitosis. Our centrosomes replicate and our microtubules begin to organize. The nuclear envelope, it is still intact and chromatin is still not condensed. So in prophase of mitosis, that nuclear material, it condensed into chromosomes. So chromatin became chromosomes. Remember, chromosomes consist of 2 sister chromatids that are connected at the centromere. The nuclear area, it's going to appear dark and heavily grainy, and the spindle fibers start to develop. During metaphase, that nuclear envelope, it's going to be completely broken down. Our chromosomes, they are going to align at what is called the metaphase plate, and the metaphase plate is not a real structure. It is an imaginary line that is at the equator of the cell. And this lining up is going to occur right before a separation occurs. Separation is known as anaphase. Anaphase is when the sister chromatids, when they have separated, and we can see them move towards opposite poles of the cell. Those separated sister chromatids, they are now considered chromosomes. Telophase, during telophase, the chromosomes, they're going to decondense to form chromatin. Overlapping with telophase, we have cytokinesis, which is defined as the division of the cytoplasm. Cytokinesis is identified by the formation of the cell plate in plants and the cleavage furrow in animals. The cell plate is the beginning of a new plant cell wall that is going to eventually separate that cell into 2 daughter cells. Meanwhile, the cleavage furrow will eventually separate the cell into 2 daughter cells and eventually pinch the animal cell into 2. So meiosis, it is actually separated into 2 different phases, right? So we have meiosis 1. This is known as reductive division. It's known as reductive division because we're going to have diploid cells dividing to produce haploid cells. And that is going to occur by that separation of our homologous chromosomes. Then we have meiosis 2. It's called equatorial division. And this is because we are going to have a mitotic-like division in which our sister chromatids are going to separate. So in interphase of meiosis, we still have that G1 phase, and what happens is that our germ cells are going to grow, they're going to perform their regular functions, we're going to duplicate our organelles, and that cell is going to start to prepare for DNA replication. Under the microscope, the nucleus appears large and lightly stained with uncondensed chromatin, so we have no visible chromosomes in G1. Then we undergo the S phase, which is known as synthesis, and we have DNA replication. Okay, so each chromosome, it's going to be copied to form 2 identical sister chromatids that are joined at the centromere. This is the only round of DNA replication before the 2 divisions of meiosis, meiosis 1 and meiosis 2. Then we end up in G2, and the cell finishes preparing for division by making proteins and enzymes that are needed for meiosis. Our centrosomes are duplicated, and the spindle fibers begin to form in the cytoplasm. That nuclear envelope remains intact, and the chromosomes are still not visible yet. So in prophase 1 of meiosis, this stage is really similar to prophase of mitosis, and I say that because the cell is preparing for division. The difference is that in mitosis, the homologous chromosomes associate with each other by lining up point for point. This pairing of homologous chromosomes is termed synapsis. They're going to form bundles of 4 chromatids that are known as tetrads, right? So we have 2 right there, and then 2 together, which form our tetrads. During synapsis, crossing over occurs. Crossing over is the exchange of equivalent pieces of 2 non-sister chromatids. So this yellow where the red is and this red where the yellow is, that is crossing over. After crossing over occurs, each of these 4 chromatids, they are going to be genetically different. And this represents a really great source of genetic variety for the species that reproduce via sexual reproduction. Our spindle fibers, they attach to the centromeres and they begin to position the tetrads. So in metaphase 1, those tetrads, they start to migrate to the equator and they line up independently one of another. This random alignment, it is referred to as that law of independent assortment, and it's another source of genetic variety because there will be so many ways possible for those tetrads to align. During anaphase 1, we have homologous pairs of chromosomes that are going to be separated. One chromosome, it's going to consist of 2 chromatids. So remember, this is one chromosome. This one chromosome has 2 chromatids. So each homologous chromosome is going to move to one pole while its homologue, its partner, is moving to another pole. So this one moves this way and this one moves that way. So we have one set of chromosomes that moves to one pole and one set that moves to the other. This separation of homologous pairs is referred to as that law of segregation. Then we have telophase 1 and cytokinesis 1. At this stage, our chromosomes, they fully reached the poles, and then cytokinesis began. In our animal cells, this is going to be called a cleavage furrow. In plant cells, it's going to be called a cell plate that's going to develop. So when cytokinesis is complete, we now have 2 genetically different haploid cells that resulted. So in prophase 2, this stage is the first stage of meiosis 2, which is equatorial division. And prophase 2 is really similar to prophase of mitosis, in which we have spindle fiber formation and so on and so forth. In metaphase 2, the chromosomes, they each consist of two sister chromatids, and they still migrate towards that equator of the cell, which is the metaphase plate. In anaphase 2, those sister chromatids, now they are separated and they're going to pull to opposite poles, and the separated sister chromatids are now called chromosomes. In telophase 2 and cytokinesis 2, those chromosomes reach the poles, and cytokinesis begins in the same fashion as in telophase 1. So when cytokinesis is complete, we have 4 genetically different haploid cells result. So understanding chromosome versus chromatid and all of that is really important. So when we're looking at this image, what we have here is 4 chromosomes. Okay, so this is a chromosome, that's a chromosome, that's a chromosome, and that is a chromosome. So we have 4 chromosomes, and chromosomes, they are known as single DNA molecules. When we have a duplicated chromosome, it's a chromosome that has an extra copy of itself to prepare for division. So when we have them like this, each chromosome is going to consist of 2 sister chromatids that are going to be attached at the centromere. So we have 4 chromosomes, but we have 8 chromatids, right? Because this is a chromatid, this is a chromatid, that is a chromatid, that is a chromatid. So we have 8 chromatids and 4 chromosomes. So when we're in that S phase, every single chromosome is going to replicate its DNA and then consist of 2 sister chromatids. As long as those chromatids are connected to one another, they are considered a single chromosome. It is not until we separate those chromatids that they're pulled apart from one another that they're going to become full-on chromosomes by themselves. So when we're looking at this, what we see in this top left image is this is most likely anaphase of mitosis. What we see is sister chromatids that have been pulled apart, moving towards opposite poles. So each chromatid is now considered a separate chromosome. So if we have 1, 2, 3, 4, 5, 6, 7, 8, we have 8 chromosomes and also 8 chromatids. When we move to this image, what we see is likely metaphase of meiosis. So we have chromosomes, these homologous chromosomes, they are aligned at that metaphase plate, and each is going to be composed of 2 sister chromatids. So if we have, that's very dark. If we have 4 homologous chromosomes, we can count the amount of sister chromatids that we're going to have. We have 1, 2, 3, 4, 5, 6, 7, 8. So we have 4 homologous chromosomes and 8 chromatids. And we know that this is going to be meiosis because we had crossing over that occurred. Crossing over is only ever going to occur in meiosis. It will never ever occur in mitosis. So when we're looking at this bottom image, again, we see that we had crossing over occur, but we only have 2 homologous chromosomes, which means we have 4 sister chromatids. So we are probably in metaphase 2 because we see that they're lined up at that metaphase plate. So when we have diploid cells, we call them 2N, and that's because they have two complete sets of chromosomes. We have a yellow and a red chromosome in a cell, and that is not enough to be diploid. Those chromosomes also have to be the same length. So when we're looking at this, we see that the cell is diploid because we have two matching pairs, two red and two yellow. There is a short red and a short yellow, and then we have a long red and a long yellow. When we're looking here, what we have is a haploid cell, which is just known as N. When we look at it, it may first appear like that cell is diploid, and that's because there's a chromosome of each color, but it's not diploid because it is not a matching pair. Oh boy, so challenge yourself. So here we're going to be comparing prophase. In mitosis, we will never ever have crossing over or synapsis. Whenever we're looking at something like this and we see that we have red and yellow together, we are going to know that it's automatically going to be in meiosis. So when we are in prophase of mitosis, what we have is condensed chromosomes, and we do have duplicated DNA. When we are in meiosis 1 of prophase, what we have is synapsis and crossing over occur. And then when we are in meiosis 2, we have half as much genetic material as we have in meiosis 1. In metaphase of mitosis, we have all of those chromosomes align at the metaphase plate, but again, we had no crossing over and we had no synapsis, so we can only be in mitosis. Then when we move on to meiosis, we have 4 homologous chromosomes. Well, we have 2 pairs of homologous chromosomes, which makes 4 chromosomes. They are diploid in this condition. And then in meiosis 2, we have individual chromosomes that are going to be lined up the mirror, but they are going to be haploid because now we only have half the amount of DNA. So once we reach anaphase, that our chromosomes, they have separated, alone, when we have them like this, they're going to be considered individual chromosomes at that point. So we had that separation of our chromatids, and now they're considered individual chromosomes. We move on to meiosis 1, and what we have is homologous pairs that are separated, and this is currently diploid. But in meiosis 2, we're now haploid because we have half of that genetic material, and we did have crossing over and synapsis occur. When we're looking at telophase and cytokinesis, what we see is that in mitosis, we have 2 identical daughter cells. When we are in meiosis, we have 2 non-identical daughter cells, and then those 2 non-identical daughter cells, they're going to separate again so that we can achieve a haploid condition. So then in meiosis 2, we're going to have 2 non-identical daughter cells again that are going to also be haploid. So this is what mitosis looks like in an onion root tip. And here we have all of the phases. Here in prophase, we see that we are starting to get condensed chromatin. In prometaphase, we see that our chromatin, they are officially condensed. In metaphase, we can see that they're lining up at the plate, and in anaphase, they're separating. And then in telophase, we see here that cell plate forming, which is going to be dividing that cell for good. This is another example of mitosis in a plant. And here we have mitosis in an animal cell. Specifically, this is a fish blastula. So here we have prophase, we have uncondensed chromatid, we have metaphase here. We have all of our chromosomes lining up at that metaphase plate. We have anaphase, which is separating those sister chromatids. And then we have telophase, which is that separation of the cell. Oh, and here we have interphase, in which all those chromatin are really diffuse and granule.